Hey, what's up everybody, it's Roy from India. Welcome to my channel Roy's Academy. Make sure you watch the video to the end, because if you don't, there might be some punchlines at the end of the video which is very much important for clearing the concept. And also at the end of watching the entire video, please do hit the thumbs up button and also comment down in the comment section below and let me know if you have any personal queries or doubts. And also you can show me your love and affection by subscribing my channel, so please do subscribe my channel and help me grow my channel more and more for you people, so that I can help benefit you people with more such videos. Thank you. So today I'm going to start with protein metabolism. So actually there are general pathways, different pathways on the protein metabolism aspect. Okay, purine metabolism is there, pyrimidine metabolism is there, and then urea cycle is there. Different amino acid metabolism is there. Then the nucleotides, different nucleotides, the purine nucleotides and the pyrimidine nucleotides separately, different kinds of metabolism are they presented. So in this case, what happens? Protein metabolism. In protein, there are metabolism has two pathways, as we know, right? One is the synthesis and another is the catabolism. Now in synthesis, what happens? From amino acids, the higher groups of proteins are being synthesized. And in case of the catabolism, from the larger polypeptide chains of the proteins, the amino acids are being synthesized or formed, and lastly the amino acids are broken down to ammonia, which are being converted by the urea cycle in the liver and then excreted out of the body, right? Now this ammonia takes the help of another molecule which is called CO2. So where does this CO2 come from? This CO2 is basically coming as the end product of cellular respiration. Okay, the end product of the cellular respiration. Now this ammonia plus CO2 in the presence of carbonyl, phosphate synthase 1 gets converted to what? Carbonyl phosphate. So basically this ammonia is coming from where? The breakdown of the proteins, right? This ammonia is basically coming from as the end product of protein catabolism. Okay, like I said, the different kinds of proteins are being catabolized to amino acids, and then the amino acids are broken down to what? The ammonia, lastly. This ammonia, so what does the basic function of this urea cycle? What is the basic function of this urea cycle? Urea cycle does nothing but converts this ammonia to less toxic form through the urea cycle and forms the urea, which happens specifically in the liver. Okay, and excretes it out of the body through the urine or sweat, and some quantities are also excreted out of the body in the form of stool or the excretory product, right? Now this carbonyl phosphate, takes the help of L-ornithine. L-ornithine is actually a byproduct of what? Arginine. How this ornithine is coming, I'm coming right back to it. But first, coming to ornithine, how the ornithine comes through the cycle of carbonyl phosphate, and how the carbonyl phosphate takes in action of ornithine is needed to be incorporated here. So how does it incorporate the actions of L-ornithine? That is very much important. So the carbonyl phosphate in presence of ornithine, L-ornithine, gets converted to L-citrulline. L-citrulline. Okay. Now this L-citrulline compound, in the presence of another enzyme which is called ornithine transcarbamylase, ornithine transcarbamylase gets converted to L-citrulline. Now this L-citrulline in presence of another enzyme arginosuccinate synthase. In the presence of another enzyme, arginosuccinate synthase converts to arginosuccinic acid. And this conversion takes place in the presence of Mg2 plus ion, which gets converted to Mg, and also ATP. ATP gets converted to AMP, adenosine monophosphate, plus PPI. So the inorganic phosphate, the breakdown of the ATP to AMP, it's an energy generated active process. But the ATP is converted to AMP and PPI. In that case, what happens? This broken energy is being taken up by this arginosuccinate synthase in order to decrease the activation energy of L-citrulline. Now, when this energy is being incorporated by the arginosuccinate synthase enzyme, it generally helps in getting more active energy to decrease the activation energy of the del G value, the Gibbs free energy value of the L-citrulline, and converts it to the arginosuccinic acid. Now this arginosuccinic acid in the presence of arginosuccinate lyase, lyase means the enzyme which splits a compound into 2 different or 3 different other products or compounds. So the arginosuccinate lyase breaks down the arginosuccinic acid into fumarate and also forms L-arginine. So the carbonyl phosphate, which was formed from the ammonia, which is the broken end product of the protein molecules, which converts to amino acids, and amino acids are leaving behind due to the action of the enzyme or the conversion of the more toxic form to the less toxic form in the liver. It gets converted to ammonia, the end product of the protein metabolism, or as a byproduct. Now this ammonia, with the help of the CO2, which is being released by the Krebs cycle or the glycolysis or by the conversion of the payoff conversion of pyruvic acid to the acetyl CoA, it leaves the CO2 molecule, and this mostly in cases of the cellular respiration, where the CO2 molecules are being produced as the end product. In those cases, what happens? This CO2 and the ammonia gets combined with each other in the presence of carbonyl phosphate synthase 1 and gets converted to carbonyl phosphate. Now the carbonyl phosphate, in the presence of ornithine transcarbamylase and with the help of or aid of L-ornithine means levorotatory ornithine, gets converted to L-citrulline. The L-citrulline compound in the presence of arginosuccinate synthase gets converted to arginosuccinic acid. Now arginosuccinate synthase takes the aid of cofactor Mg+2+ or Mg++, which is a metallic cofactor, which helps in transferring the electron group or the radical group from one part of the compound or the substrate to the another part, and it aids in activating or catalyzing catalyzing the enzymatic reaction more successfully. Now what happens? This ATP here converts to AMP and PPI, adenosine monophosphate and inorganic phosphate, two inorganic phosphates or biphosphates. With the help liberation of this energy from the ATP to AMP and PPI, that energy is utilized by arginosuccinate synthase in order to bring about the lowering of the activation energy of Lcitrulline, which is the substrate, and after lowering the activation energy of the Lcitrulline, it gets converted to arginosuccinic acid. Now the arginosuccinic acid in presence of arginosuccinic lyase gets converted to L-arginine, and also lyase splits down the arginosuccinic acid to fumarate. Okay, so fumarate here is a byproduct. So this fumarate is getting converted to fumaric acid, or it acts as the electron donator in case of the electron transport chain, or the fumarate leaves out of the body as an excretory product through the urine, but in very less amount or less quantity. So fumarate or the fumaric acid, whatever it is, it's get converted. It's a very less amount of toxic byproduct that has been formed in this entire urea cycle. Okay, now this L-arginine in the presence of another enzyme called arginase. Arginase gets broken down into two different products. One product is urea and another one is L-ornithine. Okay, so this urea is CONH2 is the most less toxic form of ammonia that gets excreted out of the body in the form of urine or through the sweat, or in sometimes through the stool too, in the form of stool. Okay, or through the different means of excretion that happens through our body. And this urea not only happens in this urea cycle, this is called urea cycle. It doesn't only happen in case of the human body, but also in most multicellular organisms' body, specifically mammalia group. Those are animals which have consumption of proteins, or which has the capability of metabolizing or catabolizing, synthesizing proteins and amino acids in their body. Okay, so the urea cycle mainly happens in case of the mammal group, unlike the aves and the reptilia group. So we are ureotelic, and in case of reptiles, this urea again gets converted in case of the reptiles, or in case of the lower group of multicellular organisms, the terrestrial animals or the amphibians. Okay, this urea gets converted to uric acid. Okay, now this uric acid is being excreted out of the body with a form of urine or different other pellet-like or droplet-like structure. And this, apart from this urea and uric acid, this uric acid also is the form of the excretory product substance in case of birds, the group of aves. Okay, apart from reptilia and also amphibia. And ammonotelic, there is also ammonotelic. Ammonotelic organisms are specifically the higher group of mammals like humans. So in humans, not only urea is excreted, but also ammonia is excreted. In bears, in case of the lions, the carnivorous animals which feed on the proteins, specifically muscle proteins of other herbivores. Okay, so in our cases, or omnivores, like bears and chimpanzees, primates, gorillas, apes, then wolverines, civet cats and all these kinds of organisms in mammal group who are carnivorous in nature, they specifically feed on protein. In those cases, they are also ammonotelic, that is excrete ammonia out of their body through the form of stool or fecal matter, fecal wastes. But also in their body, the urea cycle, this cycle more or less happens and converts it to less toxic form, that is urea. And this urea gets ejected out of the body through the urine or sweat and also through the form of fecal matters. Right? So this is all about the urea cycle.